In this section, we're going to look at how we take the genetic information off of the chromosome, is the genes, and use it to make the proteins. Genes are going to specify what your protein is via transcription and translation. So early evidence from the study of metabolic defects led us to realizing that the genes were going to dictate the phenotypes. They looked at nutritional mutants in the neurospora. Beetle and Tatum were the ones that originally came up with this one gene, one enzyme hypothesis. In 1958, they discovered that the genes are going to actually act by regulating definite chemical events. The product of gene expression has been revised. It was revised to the one gene, one polypeptide hypothesis, which is still not entirely correct. What we now find is that many eukaryotic genes are going to actually code for a set of closely related polypeptides due to alternative splicing. In studying the human genome and sequencing the human genome, this was a big contributor to this knowledge. They originally thought humans had about 100,000 genes. We're now down to the idea of estimating humans have about 10,000 genes. There are a few genes that are going to code for Rna molecules, and the Rna molecules themselves have important functions in the cells, and they never are translated into proteins. The basic principle of transcription and translations is that DNA is going to provide the instructions for making proteins, but it does it through RNA. If we look here at this prokaryotic cell, you've got transcription, which is going to be the process of taking the DNA, and we're going to make a copy of it as an mRNA. That mRNA will then have the ribosome attach and translate the code to make the polypeptide. All of this happens in the same compartment in a bacterial cell. You can actually have the ribosome hop on here and start reading this mRNA before it's finished, because it's all in the same compartment. With the eukaryotic cell, it's going to be a little bit more complicated. You're going to have your DNA surrounded by the nuclear envelope in the nucleus. It will undergo transcription to make your pre-mRNA. This is going to undergo some additional processing to finally become your mRNA. The mRNA will then be sent out into the cytoplasm, where the ribosome will attach and translate the code to make the polypeptide. If you think of it like a simple copy machine here, with the prokaryotes, you're going to be making copies. As they start to feed out the copy machine, you can grab the papers out of the pile and start using them to do whatever project you're doing. With the eukaryotic cell, here you're not allowed to access those papers until the whole pile is copied. And then it's going to modify it. It's like hole punching it and stapling it. Then you have the finished product get sent out. Transcription is just the synthesis of RNA, and it uses DNA as a template. Your mRNA is your messenger RNA. This is the gene's protein building instructions. This is going to be what carries the genetic message from the DNA to the protein synthesizing machinery of the cell, which are your ribosomes. Ribosomes are just your complex particles that will facilitate the orderly linking of the amino acids into the polypeptide chain. They're considered your protein synthesis organelle. They're going to make sure that each of those amino acids is put in the chain in the correct order. Translation is this process of synthesizing the polypeptide using the information from the mRNA. So slight differences in eukaryotes. Your transcription occurs in the nucleus and translation in the cytoplasm. In prokaryotes, you don't have any compartments, so it allows translation to begin while transcription is still in progress. The eukaryotic RNAs are modified before they leave the nucleus to form your final functional RNA. The pre-mRNA is that initial RNA that's transcribed prior to modification. It's sometimes referred to as your primary transcript. When we look at the genetic code, you have only 4 nucleotides to code for 20 amino acids. So you can't just use one nucleotide to represent each amino acid. A combination of 2 would be 4 times 4 possible combinations, or 16, still not enough. So we have to use combinations of 3 nucleotides, which gives us 64 possibilities to have enough. It's a triple code. Your genetic instructions are written in the DNA as a series of 3 nucleotides. And that's going to be translated into 3 nucleotides in the mRNA. And we call these the codons. So here's your DNA template. You're going to use transcription to make your mRNA, and during translation, you're going to have the ribosomes read these three base pairs at a time that form a codon. So here, this UGG is going to code for an amino acid. This UUU will code for the next amino acid, GGC for the next, UCA for the next one, and so on. This is your DNA template strand that's going to provide the pattern or the template for the sequence of nucleotides in the RNA transcript. When we look at any given gene, the same strand is always used as the template every time. But other genes that are on the same DNA molecule may be on the opposite strand. When we look at RNA pairing, it's going to pair similar to DNA, but it will substitute uracil for thymine. So any place where you would have thymine inserted in DNA, you would insert uracil to make the RNA. It's also going to be synthesized in anti-parallel directions to the template strand of DNA. Your codons, these are the mRNA nucleotide triplets that code for specific amino acids. They're customarily written in the 5' to 3' direction because they are read by the translation machinery in the 5' to 3' direction. So we have 64 codons. 61 of them are going to code for amino acids. 3 of them will be stop codons that mark the end of translation. It's important that you establish the reading frame, and AUG is going to be how we do that. AUG codes for methionine, and it's the start. Later on, you may have an enzyme subsequently remove the methionine from the chain since they all start with methionine, but that is how we mark the start. Just like when you start reading a sentence, you look for a capital letter. What you'll see is there's redundancy in the code, but there's no ambiguity. Your reading frame is going to give you the ability to read the correct message, and it depends on reading the symbols in the correct groupings. It would be just like if you took a sentence and you took and removed all the spaces from the words. If you put the spaces in different places, the words would have different meanings. It's the same thing with the genetic code. We do find that the genetic code is nearly universal, which does allow genes to be transplanted between different species. There are a few exceptions where there are translation systems with a few different codons other than the standard ones. You see some slight variations in a few of the unicellular eukaryotes and the organelle genes of some species, but by and large, nearly all organisms are going to use the same genetic code. So this is a codon table. This would be how you determine what things are going to code for. So if we start with AUG, when you see there's an AUG, your first letter is A, so you look in this first column here on the left. The second letter is U, so you're going to find that across the top. When you bring that down, it's going to take you to a box. Over on the right side, you find the third base, G. So where all of those lines cross together, it should take you to the AUG. And over here, it's going to give you the name of the amino acid or the abbreviation for the amino acid that's going to code for. So AUG codes for MET. If you had something that coded CCG, you would go C over here, C over here. When you come down to the box, you find the G over here, CCG. It's going to code for proline. If you had CGA, you could look over here, C, G, it's going to bring you down to this box, A, it's going to code for arginine. If you have UAA, that's one of them that codes for a stop. There are the 3 stop codons. That's going to tell you when you've reached the end of the gene. It's like having a punctuation mark at the end of a sentence. When we look at the molecular components of transcription, you're going to use an RNA polymerase here. When you're just copying the DNA, it was a DNA polymerase. Here we use an RNA polymerase because we're actually building an RNA polymer. It's going to split the 2 strands of DNA apart and join the RNA nucleotides together in the 5' to 3' direction. This is also going to need a primer to start. Your promoter is going to be a DNA sequence where the RNA polymerase can attach and initiate transcription. The terminator is the sequence that signals the end of transcriptions. It's slightly different in the prokaryotes and eukaryotes. Your transcription unit is going to be the stretch of DNA that gets transcribed into the RNA. In bacteria, they have just a single RNA polymerase that they use to make all types of RNA. The rRNA or ribosomal RNA, the mRNA, any of them. Eukaryotes have at least 3 different types of RNA polymerases. Your starting point when you go to make this RNA transcript is going to be the nucleotide where RNA synthesis actually begins. Your promoter is usually several dozen nucleotides upstream from the start. Here in this picture you can see you've got a starting point, and then you've got the promoter here, and the RNA polymerase will attach. Transcription factors, these in eukaryotes are a collection of proteins that mediate the binding of the RNA polymerase and the initiation of transcription. Only after you have the transcription factors attach and the RNA polymerase II bind will it begin. In prokaryotes, the RNA polymerase is going to bind specifically and recognize the promoter. Your transcription initiation complex is going to be the complex of transcription factors, the RNA polymerase II, and being bound to the promoter. Within the promoter, we've got this sequence we refer to as the ta-ta box. It's going to contain TA, about 25 nucleotides upstream from the transcription start. Here you can see you're going to have your RNA polymerase attached to the promoter. You've got your transcription unit. It's going to unwind the DNA and allow you to start to make this RNA transcript. It will eventually elongate as this progresses down this DNA template. After it passes through, it will allow the DNA template to rewind, and you'll end up with your double-stranded DNA still being intact and your completed RNA transcript. The interaction between the eukaryotic RNA polymerase II and the transcription factors is an example of how important these protein-protein interactions are in helping to control transcription. RNA polymerase is going to untwist the helix and expose about 10 to 20 nucleotides at a time. It adds nucleotides to the 3' end, and then the RNA is going to pull away from the DNA template and the DNA helix will recoil. This occurs at a rate of about 40 nucleotides per second in the eukaryotes. A single gene can be transcribed simultaneously by several RNA polymerases. So once one RNA polymerase is done, another one can hop on behind it, so you can make several copies of the gene to send out to the cytoplasm and to the ribosomes. The termination mechanism will differ slightly in the eukaryotes and prokaryotes. Bacteria going to have transcription proceed through a terminator sequence in the DNA. That transcribed terminator itself functions as a termination signal. In eukaryotes, the RNA polymerase II will transcribe a polyadenylation signal sequence. Sequence is AAU-AAA, and this will be in the pre-mRNA. 10 to 35 nucleotides downstream, it will cut free the pre-mRNA. Eukaryotic cells are going to process and modify the RNA after it's transcribed. So RNA processing is just when you take that pre-mRNA and modify it before the genetic message is actually dispatched. Both ends are altered, and in many cases some of the interior sections are also going to be modified. So when we look at how we alter the ends, you've got a 5' cap where a modified form of guanine gets added onto the 5' end after transcription of the first 20 to 40 nucleotides. At the 3' end, you're going to have a poly-A tail. Here, an enzyme will add 50 to 250 more adenine on there. This 5' cap and poly-A tail seem to play a role in exporting the mRNA from the nucleus. They help to protect the mRNA from degradation by hydrolytic enzymes, and they help the ribosome get attached to the 5' end of the mRNA once it gets into the cytoplasm. So RNA splicing is a big way that we modify genes. With RNA splicing, you remove large portions of the RNA that were initially synthesized. The average human transcription unit has about 27,000 nucleotide pairs. Only about 1,200 nucleotides are in the average protein of 400 amino acids. So a very significant portion of it is cut off. Here you've got your pre-RNA, you've got a 5' cap and a poly A tail. We have these regions called introns and exons. The introns are going to be these intervening sequences. They're non-coding segments of the nucleic acid. The exons are going to be the expressed regions. They're going to be the ones that you're going to splice together. So here you've taken this 3 sections here and connected them together to make your actual coding segment. SNRPs, or small nuclear ribonuclear proteins, are going to be short nucleotide sequences at the ends of each intron. They're used as a signal for RNA splicing. So these introns, in a lot of cases, are going to have their own message to splice them out. So the small nuclear RNA is going to be the RNA in a SNRP. Each small nuclear RNA is about 150 nucleotides long. A spliceosome is going to be several SNRPs that are joined with additional proteins. These end up interacting with additional sites along the intron, and they'll release the intron to be degraded and join the exons. So small nuclear RNAs will catalyze this and then participate in the spliceosome assembly and splice site recognition. So in this example here, you've got this giant intron in the middle. The SNRPs are going to come along with your small nuclear RNA, and they are going to help to facilitate pulling this out and joining up these segments so that you can have this cut out intron and the exon segments being joined together. Ribozymes, these are RNA molecules that function as enzymes. So in some organisms, the RNA splicing occurs without proteins. The introns function as a ribozyme and catalyze their own exclusion. RNA does work well as an enzyme for a few reasons. It's single-stranded, so this allows complementary base pairing and gives it a 3-dimensional structure that is essential for catalytic function. Some of the bases in RNA will have functional groups that may participate in catalysis, and RNA has the ability to hydrogen bond with other nucleic acids and add specificity to the catalytic activity. The introns are important. While they may sound like just a region that's being cut out, they are far more important than that would suggest. The single gene may encode more than one kind of polypeptide. Many genes give rise to 2 or more polypeptides depending on which segments get treated as exons. And this happens through alternative splicing. So if we go back up and look at this piece here, with alternative splicing, you may take this first and second section. You could take the first and third section. You could take the second and third section. You could take all 3. Those would be all alternative ways that this could be spliced together. So alternative splicing gives you 2 or more different polypeptides. Your domains are the discrete structural and functional regions of the modular structural proteins. You may want to have several of the same domains and then just alter different regions in between. With exon shuffling, your introns increase the probability of a crossing over event occurring between the exons of an allele of a gene, and that would result in new combinations of exons and proteins. Now we'll look at translation. In translation, the cell reads the genetic message along an mRNA and uses that as the instructions to make a polypeptide. Your tRNA is your transfer RNA. It's going to transfer an amino acid to a growing polypeptide in the ribosome. It acts like a translator, so each tRNA will translate a particular mRNA codon into a specific amino acid. I like this picture on the end here. You've got your tRNA here. On the very end is where you would attach the amino acid. And then down at the bottom, you've got these 3 base pairs here that make up the anticodon. It's a nucleotide triplet at the opposite end of the amino acid, and it's going to be complementary to the codon in the mRNA. That way you can verify on the tRNA that you've brought the correct amino acid by making sure you're using the complementary sequence to piece them together. So they're going to have some single-stranded RNA on them, and they're only about 80 nucleotides long. Your amino-acyl tRNA synthases are the family of enzymes that carry out the matching up of the tRNA and amino acid. So the correct amino acid must bind to the tRNA. So you have 20 different amino acids, 20 different synthases, one for each amino acid, and each is able to bind all the different tRNAs. Your synthase catalyzes the covalent bonding of the amino acid. It's going to bring its own energy through the hydrolysis of ATP. Your tRNA anticodon has to pair with the appropriate mRNA codon. So what we would expect is if one tRNA existed for each codon, we would see 61 different tRNAs. But what we see is there's about 45 of them, which suggests some of the tRNAs combined more than one codon. This is due to what we call the wobble, or the flexible base pairing at the third position of the 3' end of an mRNA codon. The rules for pairing the third nucleotide of a codon and the corresponding tRNAs are more relaxed than in other positions. So if we go back up here and look at our codon table, we look, for example, at valine here. You have GUU, GUC, GUA, GUG. As long as you have GU as the first two, it doesn't really matter what's in this third position. It's going to code for the same thing. If you look at alanine, you see the same thing. Proline, you see the same thing. So this is what allows for that wobble. Your rRNAs are the ribosomal RNAs. They're the subunits of the ribosome. So part of the structure of the ribosome is RNA itself. The ribosome is going to be the organelle that facilitates the coupling of the tRNA anticodon with the mRNA codon. They have a large and a small subunit that are made up of proteins and one or more RNAs. Bacterial and eukaryotic ribosomes are similar in structure and function, but different in size. So the eukaryotic ribosomes are going to be larger and slightly different. Certain antibiotics attack prokaryotic ribosomes. This will allow them to attack bacterial cells without affecting eukaryotic ribosomes. Drugs like tetracycline and streptomycin are going to do this. Each ribosome is going to have 3 binding sites for the tRNA. Here we look at it, you've got the P site, which is your peptidyl tRNA binding site. Or the easy way to remember it is where you're going to hold the growing polypeptide. That will be right here. This is where your growing chain is present. Just in front of it, you're going to have the A site, the aminoacyl tRNA binding site. This is going to accept the tRNA. This is going to accept the new one that's coming in, bringing the next amino acid. Your E site is the exit site. This is where you're going to have your discharged tRNA leave. So as they are progressing down the line, what is in the P site eventually moves to the E site. What's in the A site eventually moves to the P site. What's in the E site is going to be ejected. So your ribosome holds the tRNA and the mRNA in close proximity and position so that a new amino acid can be added to the carboxyl and then catalyze the formation of the polypeptide bond. So as this comes in, they're held in close proximity. You're going to form this polypeptide bond. This slides in. The tRNA is ejected to go out and be recycled. So the translation initiation complex is going to be your union of an mRNA, an initiator tRNA, and a small ribosomal subunit followed by a large ribosomal subunit. Your initiation factors are proteins that are required to bring the translation complex together. Your polypeptide is directional in how it's synthesized. It's always going to be synthesized from the N terminus to the C terminus. Your elongation factors are proteins that are going to be involved in adding amino acids to the previous ones. This will happen at the C terminus. Your release factors are going to be proteins shaped like an aminoacyl tRNA. They'll bind to the stop codon in the A site, and when this goes to slide in, it will cause the addition of water instead of an amino acid, which hydrolyzes the bond between the completed polypeptide and the tRNA at the P site and allows it to release. It's like having a blank come in that it tries to attach to, and it realizes, nope, you don't have an amino acid, you have a water instead, and then it will be released. When you have polyribosomes, you can have multiple ribosomes translate an mRNA at the same time. Here you can see this is a single mRNA and multiple ribosomes lined up. So these are strings of ribosomes or polysomes. This will allow you to make a very large amount of protein from a single set of directions. So when we go to complete the process, your polypeptide chains are going to begin to coil and fold spontaneously based on the amino acid sequence. So if you remember back to the beginning, your amino acid sequence was the primary structure of your protein. It's going to fold to give specific shapes of your secondary and tertiary structure, and that's often helped by the chaperone proteins. Even after translation, you can have some modification. Here, additional steps would be required before that protein goes on to do its job. So you can chemically modify it. It may be cleaved. It could bind to another polypeptide for the quaternary structure, for example. The free ribosomes are typically going to synthesize proteins that are going to stay in the cytosol and function there. Your bound ribosomes are going to make the proteins of the endomembrane system and your secretions. You'll have signal peptides that will mark the polypeptides of the proteins that go into the endomembrane system, and it will target the protein to go to the endoplasmic reticulum. The SRP recognizes the signal peptide, and it's going to function like an escort to bring the ribosome to a receptor protein in the endoplasmic reticulum. Here you have the SRP. Other signal peptides get used to target the polypeptides to other organelles. So in this process, some things can go wrong with mutations. Mutations of one or a few nucleotides can affect a protein structure and function. Mutation is just a change to the genetic information of a cell or a virus. This is responsible for the diversity of the genes. A point mutation is going to be just a single nucleotide pair of a gene that is going to be changed. This can have a pretty profound effect, like sickle cell disease. So our small-scale mutations we can look at here. With the nucleotide pair substitution, you replace one of the nucleotides and its partner with a different nucleotide. This can result in a silent mutation, which has no observable effect on the phenotype. It's going to translate into the same amino acid, so you'll end up with the same protein. With a missense mutation, you change one of the amino acids. It may have little effect or a major change. Sickle cell anemia is actually the result of changing just one base pair, which changes one of the amino acids, and the red blood cells will become sickle-shaped. With a nonsense mutation, it's going to cause translation to stop early. Another thing we can have happen is an insertion or a deletion. With an insertion, you add a nucleotide pair into the gene, and with the deletion, you lose a nucleotide pair. This will result in a frame shift mutation where you alter the reading frame of the genetic message. Usually this is going to be non-functional. In this section here, you can look, this is your wild-type DNA template strand, and this is going to be the mRNA and the sequence that's coded for. If you have a nucleotide pair substitution with a silent mutation, it's going to code for the same thing. Where with a missense mutation, you change one of the letters in a place that will actually result in bringing a different amino acid. With the nonsense, you would actually terminate it early. Here, it would not make the full sequence. When you have a nucleotide pair insertion or deletion, here you change the whole reading frame. So here you've stuck in an extra letter, and it's caused it to read differently. Here, with the A missing, it ends up with the U missing, and you end up with two different amino acids. Here you've got a frame shift with an extensive missense. Here there's no frame shift, but one amino acid is actually missing and you stop early. Mutagens are physical and chemical agents that are going to interact with the DNA and cause these mutations. The spontaneous mutations are created by mismatch during replication. It's estimated 1 in 10 to the 10th nucleotides is altered and changed and gets passed on to the next generation. With mutagens, some examples are X-rays, UV light, and chemical mutagens. Some of the mutagens are actually carcinogens, which are cancer-causing substances. While gene expression differs among the domains of life, the gene concept itself is universal. Bacteria and eukaryotic RNA polymerases differ significantly from each other. There's only a single Rna RNA polymerase, and it resembles the three eukaryotic ones. Your Rna ribosomes are the same size as the bacterial ones, but they have chemical sensitivities that are more similar to the eukarya. Bacteria, they can simultaneously transcribe and translate the same gene because they lack the compartmentalization. And the genes are the region of the DNA that can be expressed, and they will produce your final functional product that's either a polypeptide or an RNA molecule.